All right. Hello, everyone. Welcome to the official start kickoff to the Trek Seminar Series. Today we have Adam Herdman um, in the formerly known as Environmental Horticulture Graduate Program. Adam has always been captivated by the complex relationships on display in the natural world. During his undergraduate degree, he became involved in research on threatened and endangered orchid species in the Florida Everglades, and he became enamored with the delicate beauty of many species throughout the region. Adam has traded the cornfields of Illinois for the swamps and sloughs of South Florida, welcome, in an effort to make this interest a career. It is his hope that by focusing on charismatic species such as the ghost orchid, that a framework for native orchid conservation can be built. Adam stresses working with land managers and private stakeholders to directly inform on research goals and restoration strategy. To address regional scale analysis of the many fragmented populations, Adam has partnered with many organizations to disseminate lessons learned and operate within a greater network of plant conservation. I hope you'll help me welcome Adam Herdman to today's seminar. Thanks.
Thank you for that introduction. Let me share my screen real quick. Top. All right. Can everybody see my screen? All right. I'll get started then. Thank you all for coming, and thank you for the introduction. I would like to spend time today talking about native orchid reintroduction and specifically when it comes to my work with the ghost orchid. So conservation more generally, many of you might be aware, but some may not. In order to conserve one species, there are a lot of things that need to happen prior to that. So to conserve any plant species, it is important to engage with field monitoring of the plants in the wild. So actually getting out there, getting boots on the ground, and understanding what these populations and what the stressors are doing out in the wild. Then it is important to engage with lab propagation and research. So now that we've understood what's going on in the field, we need to study the plant where we can control conditions and better understand aspects of its biology, reproduction, genetics, etc. And then the end goal of any conservation effort is, of course, reintroduction, taking these plants and supplementing the wild populations to make sure that uh, you know natural beauty continues to exist into the future. Um, and so all of these things need to happen in order for us to conserve a species in the field. That's a laundry list, and it's made even more difficult because, of course, we often are only concerned about conservation when a plant becomes rare or endangered. So it's a species that's under a lot of threats already. And what that means is we're often constrained in terms of the material we have to work with, the populations we have to observe, and I'll get more into that later on in the presentation. Excuse me. So reintroduction of plant species, it has been my experience that for a lot of researchers, reintroduction is more of an afterthought. So we focus on collecting population or collecting from populations in the wild. We focus on propagating them, making sure they're preserved in the lab. And we do our experiments, and then reintroduction is seen more of an afterthought. We put the plant material back out there. Sometimes it's informed, sometimes it's not, but it's kind of seen as a dead end among many researchers because it's time consuming. It can take many years, as you're about to see. Um, and you can't control what happens to that plant material, of course, when you put it out into the wild. So when it comes to reintroduction, a lot of the time, as far as research goes, We're purely looking at things like mortality over time, which is a simple metric. And we could be doing a lot more by engaging more intentionally with reintroduction. So, on top of all of that, what makes orchid reintroduction challenging is epiphytic orchids grow on a host tree in South Florida, and they rely on a lot of different species operating in tandem in order for a orchid to exist in the wild. So in the wild, it um, for an orchid to be there for you to see, it relies on a healthy pollinator population. So pollinators need to be going from plant to plant. Um, a lot of um, a lot of orchid species rely on what's called a mycorrhizal fungus. It's a fungus that the plant needs to persist in the wild. It also relies on a host tree. It's a epiphytic orchid. It grows on a host tree. So no host tree, no orchid, right? And then finally, you need a sufficient population size in order for there to be a healthy amount of reproduction going on. And this is before you take into account things like temperature, humidity, and of course, wildlife that may prevent you from getting to the field sites, like, the gators. And what all of these complex factors mean in terms of the distribution that we see in the field is that um, all of our native orchid species are at different distributions. Some may be way up in the canopy, such as our jingle bell orchid, and some are very close to the water line. All of them rely on a different mosaic of pollinators, of fungus, of host trees. So this is what drew me into this field, and what I find so rewarding is to try to piece together the puzzle of what explains where these orchids are in the wild. And this is just a list of our native orchid species that are common out throughout the Fakahatchee Strand, which is where I study. Many of them might be um, known to you, but I would just like to point out here the diversity of flowering that you see, and appreciate that each one of them rely on a slightly different number of factors working together in order for them to be out there. Now, excuse me. For our native epiphytes, the ones that grow on host trees, uh, most of them are found in South Florida where the climate becomes subtropical. So there are some epiphytic species that grow up in North Florida, but most of the species throughout the United States are actually terrestrials. They grow in the ground. But only here in South Florida is where you find these epiphytic orchids. And it's a special combination of the hydrology, so having these freshwater swamps that keep it humid enough for these orchids to exist, but also that climate where the temperatures don't drop as drastically. Unfortunately, we had a big freeze this year, so we'll have to see how that goes. And this is the sort of contrasts that you would see in the Fakahatchee Strand outside of Naples. So it goes very quickly from these mesic pine flatwoods that, as everybody here might be from Florida, these ecosystems burn and they burn often. So, um, as you may guess, orchids are opposed to burning. They will not survive a burned ecosystem. So, just a change in hydrology or of a few feet allows these orchids to persist. It goes from within maybe 200 meters, it goes from the flatwoods to the cypress swampland. So these orchids really are constrained and aren't able to, create new populations that easily because, of course, orchids cannot persist in these areas where they burn. And we've known about the Fakahatchee Strand a long, long time. It's a series of swamps and sloughs that run south interior from Naples. And I just want to point out that this is something that we've known for a long time. And also, along with knowing this ecosystem for a long time, it's also an ecosystem that has been changed over time. And that's what we need to, when we're considering reintroduction, we need to take the history of the landscape into account. So this is an ecosystem that was heavily logged in the 1940s. And so when we're thinking about our native orchid species here, their distributions would have been impacted by these large old growth cypress being ripped out. So it really is quite heartbreaking to think about when you go to places that where there's old growth cypress still remaining, such as Corkscrew, Audubon. You can see orchids growing way up in the top of these cypress trees. So I think what we need to remember is how many orchids may have been removed during this logging. And you can still see the impact of this logging in the landscape today. So in the 1940s, all of the old growth cypress was logged out, as I just said. But this isn't only in regards to the cypress being logged, it's also in the hydrology. We can see it. All of these logging trams and canals were dug to be able to get that lumber out, and that impacts the water flow even to this day. So again, just reiterating that we've made some efforts to reverse the hydrology. We've put culverts under 75 and under 29, but still that is not fully returned the ecosystem to what it is. So I think in a lot of aspects, the distributions that we see, the remaining sites, are really these native orchid species clinging on to what's left, island, they're remnants of what was. And this is just a picture of where I did a lot of my field monitoring at the Florida Panther National Wildlife Refuge. I'm happy to say that in developing the framework for conserving native orchids, we've expanded into places like Fakahatchee Strand, Preserve State Park, Big Cypress, Corkscrew, and many others. And this is when we consider the landscape, we also need to look forward. So when it comes to development, when it comes to putting in highways and putting in culverts, I think it's also important to think about the future when you're looking at reintroduction as well. And this is not a historical threat. I think oftentimes we can separate ourselves from the pictures in black and white, but this is very much so a current and ongoing threat. And of course, in regards to the landscape being so close to the water line, all we need is for the water levels to come up in such a way. This is a freshwater swamp. All that needs to happen is for salt water to mix in with the fresh water, and of course, no trees, no orchids. So, after going through a little primer on the landscape and the things to keep in account as we move forward, I think if you've ever been out in the swamps or if you've ever been out at a park and you see an orchid on a branch, I think hopefully you're able to now appreciate everything that has to work in tandem for that orchid to be existing right there in front of you. So to reiterate what needs to happen before we do a reintroduction, we need to engage in field monitoring of the wild populations, which I'll go over, and then we need ex situ experiments in the lab and be able to propagate them in sufficient numbers to put them back out there. And this is not easy. You got to get out there with the gators and the wildlife where there's little cell service. If it was easy, everybody would be doing it. We wouldn't have any problems conserving our native orchids and plant species. And I guess what I would like to point out here is that there's so few of us that are able to do this sort of research. And thankfully, I'm happy to be one of those people. So to get into the monitoring of wild populations, As Monique pointed out in the reintroduction, I was lucky enough to be brought on 10 years ago, um, and we engaged with monitoring the wild population, so a lot of fragmented sites over the years. And it took 10 years in order to put together sufficient information to say exactly how these populations are going to be doing long term. So at the beginning of this, there were only 11 ghost orchids known of at the Florida Panther National Wildlife Refuge. And after 10 years of monitoring, there were 900 plants that we had recorded. And what's important to remember is they grow on host trees, right? So 900 plants growing on 208 host trees. So what happens if one of these trees goes down in a hurricane? So when we went out there in the swamps for 10 years, what we recorded is information regarding the host tree. So the orchid is not out there existing in a vacuum. It relies on a host tree, so it's important to document factors relating to its ecology, like the host tree. So looking at the species, the height to the canopy, the light coming through the canopy, the bark texture, etc. And we also looked at aspects relating to the orchid itself and its ecology. So, what does the mosaic of plant life look on the substrate? So in the picture of the ghost orchid on the right there, you can see that it's there with ferns, it's there with mosses and lichens. It's not there alone. So we documented what neighboring species were closest. We did the distance to that species, the height from the ground, and then aspects regarding the plant itself, like growth, root growth. This is a leafless epiphyte. It doesn't have any leaves, so it's pretty easy to track growth because it's just the root lengths, and then, of course, reproduction. And over time, this is what we found. So over time, it's decreasing in population, but I guess what I want to point out here in particular is that although we documented 903 plants over time, the demographic structure of these populations is bottom heavy. So a lot of seedlings being produced, but not a lot of them surviving. And uh, although there might have been 281 plants that were alive in 2022, uh, also it's important to remember that this is divided among several different populations, and not all of the plants are able to reproduce. So at these sites, we found that the flowering number of flowering plants, excuse me, was under 23% over the years. And when we projected that using a model, we found that it was overall below replacement. And that's because the demography is so bottom heavy, a lot of young plants that aren't able to reproduce. And what we did with these field methods is we then transposed them into a population located in Cuba. So a lot of people don't know that there are ghost orchids here in there's ghost orchids here in Florida, but there's also ghost orchids there in Cuba. And in a lot of aspects, the populations are completely different. So this paper was published in 2018 and kind of documented the differences. So here in Florida, you have cypress knees. It's very swampy, but there it's very dry, and it's in on karst limestone near to the ocean. And just to prove the point that these field monitoring techniques can be transposed to other species, um, we transposed them to the ribbon orchid, so another leafless species. And over time, we found that it was a similar situation where the population was declining over time. And what I just want to point out here is that this species within the same habitat had much lower population numbers and was on way less host trees. So if a host tree went down, you were losing a significant portion of the populations within a site. And same story. So within 10 generations of our monitoring, we were expected to see the populations completely die out. So um, while the ghost orchid is the one that we like to think of in term, it has that great bloom, very charismatic and beautiful. There are many other orchid species here in Florida that don't receive as much attention and are perhaps in more need of conservation. So this is just to reiterate the sensitivities of these populations. So um, within all of the populations, we split them up into different classes. Um, class 3 and class 4, the large plants, the robust plants that are reproducing, those are the ones that are most important to the populations because they are going to be reproducing, creating seedlings for the future. And of course, when we're thinking about orchids, something that might come up and often does come up is poaching. So if a person is going to go out there and poach, what are they going for? They're going for the big, reproducing, beautiful plants, and those are the most important ones to preserve. So, sorry, my mic just went out for a second. Um, so, now all of that field monitoring has been done. We understand what the populations look like in the field. What did we do to reintroduce and supplement the existing populations? So in 2014, seed was collected of the ghost orchid. It was germinated in the lab and it was studied in the greenhouse. So they were not only growing them for reintroduction, but they were also studying the desiccation tolerances. Is it going to be okay in a drought? But also how to properly grow them for the future, as you will see. But at the end of this program that started in 2014, the plants were reintroduced in 2018. So in 2018, 123 ghost orchids for the first time were put back out into their native ecosystem um, on 51 different host trees, and I monitored them using that same standard protocol that was developed over 10 years. Um, I applied that to this population as well. So at the time, these plants were, used to perfect conditions in the greenhouse. They had been in the greenhouse for several years. They were used to it being very humid and very warm. And we were taking them and we were putting them out into the very unforgiving swamps. So what we did is we took a lot of averages from our field monitoring of the wild populations, and we came up with a distribution regime, um, more or less an educated guess on what distribution we should target when we're putting these plants out there. But put simply, we mounted them on burlap so that the burlap would rot off after a while, and then we stapled them to the trees to help them attach and establish. And then over 7 years, that's 7 years of data collection, I had to go out there in the swamps and monitor the plants and see how they were doing. And again, the same standard field methods were employed. And this is part of that framework that we're trying to develop for conservation of other orchid species. And these methods can be applied to any orchid species that you see, and frankly, any epiphytic species. So any bromeliad, fern, moss that you see, you can look at the ecology in this way. And over time, we introduced, of course, at the start, we had 123 plants, and over time, um, the plants steadily died off. Um, Now, in 2025, we had 18 plants still alive. And while that might not seem like a lot, that is 18 plants that survived 2,500 days of varying temperatures, drought conditions, Um, 3 hurricanes affected the field site over the time. Hurricane Irma hit right before we deployed the plants. And of course, there are drought conditions when you put them out. So I guess what I would like to point out here is 123 plants dropped off steadily over time, almost linearly for several years. And if the die-offs were due to the disturbance or the drought, what you would expect to see is a steep drop-off, not a steady drop-off. So I think what we're looking at here is over time, it wasn't any one disturbance event or drought that killed this population, but um, the microsite factors that I'll get into. But with a steady drop off like this, it gives you really valuable metrics in terms of the ecology that you can analyze. So, by engaging with more intentional field monitoring practices relating to the orchid ecology, what we found were some interesting things. So, in regards, this is a time to event analysis. In regards to the neighboring species, what we found is that um, the neighboring species is pretty important to the overall survival over time, the probability of survival over time. Um, and the bark texture and host species are not so important. But the most important thing that I would like to point out is that moss is really important to the survival of orchids when you put them out there. And when you go to see orchids, when you're buying them at like a box store, like Home Depot, Lowe's, any garden shop, you'll often see them packaged with moss. So you might be thinking, well, duh. But in the field, the moss is there, and the thought is that it retains moisture for longer periods of time and gives the orchid a better edge of surviving. Um, so it's that. And although we found that attachment and host tree weren't as important to the survival of the orchids, I think what we need to remember is that orchids will attach to anything. So in this picture, the orchid roots are attaching to a plastic cup. So the orchid is still alive and it's attached, but is that plastic cup really going to convey a benefit to the orchid itself? Probably not. And in the field, of course, although we found that the host tree effects weren't that important, when you're in an ecosystem like this, where there's water, a meter high, if you're not attached to a host tree, you're not going to survive. So what this may be conveying is that the humidity, the location in the swamp is more important than the host tree itself. Um, and getting deeper into the neighboring species effects, we compared all of them over time. And what we found is that mosses and lichens are pretty important for the survival of the ghost orchids that we put out there. So, for example, when you compare a moss to a fern, a orchid was almost 4 times more likely to survive um, when it was placed next to a moss when compared to a fern. So what that means in terms of reintroduction is when you're putting these plants out there, you want to take the local context into account when you're putting the plants out there. So for example, with the ghost orchid, you want to select for mosses rather than ferns or bromeliads. And as I kind of got into earlier, why is moss important? In box stores, we see them packaged with like a sphagnum moss to keep the roots humid as they're being shipped around. But in the field, I think it may convey a very different benefit. So the humidity that is trapped in the moss, the bryophyte layer on the bark, may be the thing that's conferring a benefit to the orchid itself. Or it may be a relationship with the fungus. So the humidity that's kept in the moss may confer a benefit to the orchid, but it also may confer a benefit to the fungi, allowing it to survive or be more active for longer periods of time when the swamps dry out in the winter. So this is sort of a future direction, food for thought, um, but I think this is absolutely important to orchid reintroduction. Um, and we also monitored flowering over time. And while we had a lot of flowering over time and we observed seed capsules start to develop, in other words, the plants were pollinated. We put them in the right place where the right pollinators were. Plants were pollinated, but um, the capsules never made it to full development. They withered away before they were fully mature. Um, and so if we were not able to establish a population that was self-sustaining, so we had a population with 18 plants that survived over 2,524 days. That's great, but if they aren't able to reproduce, is it really a self-sustaining population? Was it successful? And I think it really depends on how you look at it. Um, while we did put plants out there and while we did monitor them and invest a lot of time in looking at these plants, it provided us a lot more information about the species ecology that wasn't really documented before. And it also trained an entire pipeline of researchers. So not just my experiences out there, but also many undergraduate students had internship opportunities, and of course, it's led me to where I am now. But also, when you put, even if 18 plants had survived over the, um, 7 years, those are 18 plants that weren't there before. So it's a established population that's able to persist. We just don't know enough about these populations to um, make sure that they persist long term. So we're really at the cutting edge of understanding the ecology of these species. So preparing for the future, looking ahead, this is a plant that I took a picture of actually yesterday. We're growing them in the greenhouse again at University of Florida. And we're preparing with the information that we've gathered, we're preparing for reintroduction in the future. And based off of the things we learned from our reintroduction in 2018, we are hoping to look at things like attachment, um, you know, mycorrhizal fungi, host tree, substrate, the population genetics, and of course, different field sites. Um, and it's also led to um, with engaging in these intentional field monitoring strategies, it's informed on the future research for the species. So this is actually one of my dissertation chapters that we're hoping to get results back soon for, but we targeted I think it was 7 or 8 different populations. um, it ended up being across Southwest Florida, and we went out there and we sampled for the genetics. And hopefully, what that means in the future is that we're able to select for different genetic strains, maximize diversity, and solve that problem of reproduction with our reintroductions in the future. And then also what I'm doing, this is another chapter of my dissertation, is we're looking at the um, temperature tolerances of orchid seed. So orchid seed doesn't have any endosperm with it. It's just released on the wind, and it relies on pure chance to land in the right place at the right time. And what I'm hoping to do is look at one of the factors that may impact germination. And what we found is that um, at lower temperatures, the orchid seed will not germinate, but it's still alive. Then at some temperatures, you'll have, the maximum germination. You'll have the sweet spot range for germination, so that the plant will land in the right place and get going quickly. And then, of course, at the higher temperatures, the seeds will die. And why is this important? Well, with orchids that are releasing, different species release seed year round, or in the winters and in the summers. And what happens if you have a cold snap and there's seed being released at the time? What happens if there's a heat wave while orchid seed is releasing? I mean, that could impact the distribution that we see in the wild. So that's what we're hoping to do there. And that's really all I have for you today. But yeah, if for as far as the reintroduction goes, if you have any questions, I'm happy to answer them here, or you can read the paper that was actually just published last week via the QR code on screen. I am also able to answer any questions via email if you would like. Any questions? Gonna stop sharing my screen.
Hey, Adam, I think we can wait just one more minute in case somebody's busy typing
Yeah,
something out.
no problem. Did I make good time, Monique?
You have great timing. I think this is adequate time for questions, should any arise. And if not, then everybody gets 19 minutes back of their time. All right. I'm guessing no questions, so we can end the chat. I'll stay on just for a few more minutes in case somebody has a question. So there is a question in the chat. Anything special for the ghost orchid?
When compared to other species, or I guess so when we're talking about reintroduction, I think there are several things that are special about the ghost orchid, but I think you can still apply the same framework. So, of course, the ghost orchid is a leafless epiphyte. So your monitoring will have to be slightly different when you're tracking growth. Um, but in terms of the distribution and the ecology that observed over time, you could apply that same framework and get the same sort of information on the distribution and how the population is doing. So while the ghost orchid is special, um, I think there's no reason this framework can't be applied to other species. Um, I think the only difference, of course, is going to be funding. Kara says, "Thank you, Adam. This was a great presentation. Is there ever an opportunity to help during any of your monitoring? So normally, no, but we're starting to open up workshops over the summer. We're hoping to bring as many people together as possible to share ideas. And I think there's going to be opportunity to join and see the plants that I was talking about in this presentation. If you would like, just reach out to me and I'll put you down on the list. So another comment. When you transport the seedlings from UF in Gainesville to the Strand, how do they travel? Box truck, air-conditioned cab, or special humidified boxes? So when we were transporting them to the Fakahatchee Strand, what we were hoping to do is to inoculate them with the fungi. So it was actually kind of funny. They were in a box, just like a regular plastic tote filled with water, and we put the fungi in there with them. So it was kind of like a ghost orchid soup, uh, marinating as it came down to the swamps. Not a lot of the fungi was picked up, but we wanted to keep them really moist, um, not necessarily wet, but um, they were close to water for uh, I guess 4 hours. No special travel, just in the back of a university vehicle. So another comment, did you mention the percentage of seed, which was viable? So um, I don't think there were percent viability tests on the original seed batch because it was like one capsule that they were all germinated from. So they were limited on um, resources and didn't want to dedicate it to a tetrazolium test because that kills the seed. Um, but what I can say in terms of my own collections is that the percent viability in capsules that I've collected has been very, very low, under 20% viability. So again, another aspect that um, may suggest, inbreeding depression in the wild populations. So it would be really nice to get to all of the sites and test the viability, but until get that material and get it with regularity, how much can you really say? Another question. Do you select a location randomly on the tree for reintroduction? Um, so when we were reintroducing the plant material, we used the wild population inventories that we had taken, and then we took an average. So we did put some of the plants at different orientations to try and test that. But mostly we were putting them next to that bryophyte mat where it was thought most moisture was going to be kept over time. So we didn't talk about orientation because there were only 123 plants, and there's only so many factors you can look at over time with that sort of sample size. So um, when I guess I should mention that when this project started, it was not meant to be a long-term monitoring project. I just happened to be around and monitor what was there. So we've recorded the distribution and monitored it over time. So it was more of a salvage effort. Um, but yes, we did do different heights and whatnot.
All right, I thank you everyone for asking some great questions and elaborating on Adam's research. I'm sure he appreciates your help in preparing for his upcoming defense. We hope to hear from you soon, Adam, that everything went well. Um, and I hope that some of you will consider joining us for another seminar in the future.